Hello everyone, and welcome to the SaltyNote DNA extraction protocol. As soon as you arrive to lab, the first thing you're going to do is wash your hands, put on gloves and a lab coat, and then wipe down your lab bench with 70% ethanol. Once everything is clean, you're going to grab the 1% SDS cell lysis buffer and bring it over to the hot water bath. You'll then set the hot water bath to 65 degrees Celsius. On your way back to the lab bench, grab a cooler and fill it with ice. Once you are ready for your samples, you will remove your gloves and bring the cooler of ice to the refrigerator room. You'll then grab the 20 samples you've been assigned out of the freezer and bring it back to lab. Before handling any samples, remember to always have gloves on. Make sure all samples are directly on ice. Now we need to prep the tissue homogenizer. You can clean the tissue homogenizer using 70% ethanol and then wiping down with a paper towel or chem wipe. Now we need to grab the proteinase K and RNase A. You can go to the -20 freezer and look in the enzyme box. It's a red enzyme box on the bottom drawer for the proteinase K and RNase A. Bring these back to the lab bench and place on ice. To prepare the washing reagents for the tissue homogenizer, label 2 test tubes with H2O and ETOH, or ethanol, and then fill these with nucleus-free water and 70% ethanol. Make sure to keep a clean lab bench throughout your extraction. and make sure to use 70% ethanol if needed. Next, you're going to go over to the water bath and check if the 1% SDS cell lysis buffer is clear. If it's clear, then you can bring it back to the lab bench. If not, it may need a few more minutes. Once the cell lysis buffer is clear, make sure the water bath is at 65 degrees Celsius and bring it back to the lab bench. Now you're going to lay out all of the reagents you need for the cell lysis step. This includes the 1% SDS cell lysis buffer, 0.5 molar EDTA, and the proteinase K. You can also line up your samples in the yellow test tube rack that will be used in the water bath. Starting with a 1000 microliter pipette, you're going to add 350 microliters of 1% SDS cell lysis buffer to each sample. You should only need one pipette tip for this, but if it ever inserts into the test tube, you need to throw out that pipette tip. As you pipette into each sample, make sure to shift the samples down one row in your test tube rack so you can keep track of which test tubes you've inserted the SDS cell lysis buffer into and which ones you haven't. Now you're going to add 42 microliters of 0.5 molar EDTA to each sample using the 100 microliter pipette. Make sure to cap all reagents after you're done using them. Here you can see I forgot to cap the cell lysis buffer. You should only need one pipette tip for this step as well, although if you ever contaminate the test tube tip with a sample, you need to remove it and get a new one. Finally, you're going to add 20 microliters of proteinase K to each sample. You're going to use one pipette tip per sample, inserting deeply into the sample and then pipetting all of the proteinase K out. make sure to put the Proteinase K back on ice once you're done using it. Now you are ready to start grinding the tissue samples using the tissue homogenizer. Place the tissue homogenizer in the test tube and make 10 twisting motions, grinding the tissue down. Make sure the cells are fully disrupted before moving on to the next sample. Clean the tissue homogenizer in between each sample. Dip the tissue homogenizer in the water, give it some twists, and then move it to the ethanol, give it some twists, and wipe down before going into the next test tube. You can now throw out the water and ethanol washes used on the tissue homogenizer. Make sure all of the tissues are fully homogenized and all of the samples are in the yellow test tube rack for the water bath. Bring the samples to the water bath and invert 10 times before placing in the water bath. Set a timer for 1 hour.